Okay, so let's go ahead and get started. And I just wanted to welcome everybody. Good afternoon. Welcome to the Department of Medicine Research and Innovation Conference. My name is Sonia Flores, and I am a professor and vice chair in the Department of Medicine, and I will be moderating today's session. So before we get started with the speakers, I just wanted to remind everybody that we will have the Department of Medicine Research Day on Monday, April 4th. And it will be in person, but there will also be a hybrid format. So some people may be able to join via Zoom. We will have several talks as well as a virtual poster session, and our keynote speaker will be Clifford Rosen, and his topic will be From Student to Mentor, the road well traveled and enjoyed. And I wanted to point out to everybody that this is going to be the first time that we do this conference in person for 2 years, so please try to attend. The virtual poster session will start after 1. And if you need more information, please go to the Department of Medicine website. So again, this will be on Monday, April 4th, 2022. So now let's go ahead and get started with the speakers. So Michael, if you don't mind, stop sharing the screen. And now it is my pleasure to introduce Dr. Katharina Hopp, who is an assistant professor in the Division of Renal Diseases. And after she obtained a BS in biology and chemistry from Brescia University in Kentucky, and did I pronounce that correctly? I hope I did. She then went on to the Mayo Clinic for her PhD, and she stayed there for her postdoctoral fellowship, where she actually focused on polycystic kidney disease, which has been the focus of her research for many years after that. She was recruited here to the University of Colorado, and her research has focused now on understanding the role of immune cells on kidney cyst growth. Her talk today will address how targeting immunosuppression may modulate the pathogenetic mechanisms of PKD. So take it away, Dr. Hopp. So you're muted. And everybody write your questions in the chat box, and so that we can I can actually ask the questions for you at the end of the seminar. So you're still muted.
Can you hear me now?
Yes, we can hear you now.
Helps if you have the microphone on, right?
Yeah.
So again, thank you, Dr. Flores. My lab is studying a kidney disease, a genetic kidney disease that's called Polycystic Kidney Disease, as the title states. And specifically, we are studying autosomal dominant polycystic kidney disease. ADPKD is the most common monogenic kidney disease worldwide, with an incidence at about 1 in 500. It is clinically presented as a late onset disease, and the disease is characterized by bilateral kidney cysts that develop over time. And as this picture shows, these kidney cysts grossly enlarge the kidney, they're fluid filled, and then displace the normal functioning nephron, leading to end-stage kidney disease in the end. And the ruler in this picture symbolizes the size of a normal kidney, so it just shows how drastically enlarged these kidneys are in ADPKD patients. Beyond kidney cysts, patients also present with cystic lesions in the liver, pancreas, and seminal vesicles. And the major cause of mortality and morbidity is due to vascular abnormalities. To date, there's only one FDA-approved drug for ADPKD, and its use is limited to patients that have rather rapidly progressive disease. And the drug itself also just slows disease progression moderately. So there's plenty of room for new development and understanding the mechanisms of the disease to find new therapeutic options. Genetically speaking, ADPKD presents by mutations to 2 genes, PKD1 and PKD2. Mutations to PKD1 are more common than mutations to PKD2, and patients with PKD1 mutations also present with more severe disease, as shown by these two MRI images of two patients of the same age, where there are clearly more echolucent cysts in the kidneys of this PKD1 patient than the PKD2 patient. And with that, end-stage kidney disease is typically reached by ADPKD1 patients at 54 years of age versus 74 years of age for ADPKD2 patients. The two genes encode the proteins polycystin 1 and 2, and even though they have been discovered for 22 years now, there is really no true consensus what their key function is that drive that cystic growth within the kidney or the extrarenal organs. Beyond being a monogenic disease, ADPKD is also characterized by high allelic heterogeneity, meaning that no single mutations to either PKD1 or PKD2 accounts for more than the total. So it is not like cystic fibrosis as we know it, where there is a mutational hotspot where it is worthwhile looking at the gene and figuring out what that particular mutation does to then find a therapy. Beyond the genetic and allelic heterogeneity, ADPKD is characterized by a tremendous phenotypic heterogeneity. And here I'm showing MRI images of 3 patients that all have a truncating mutation to PKD1, though the effect on the protein would be the same, that they have a 50% dosage reduction of polycystin 1. But nonetheless, phenotypically, you can appreciate by these MRI images that this patient presents with countable, very few cysts, versus this PKD1 patient, which has innumerable cysts within their kidney at the same age. There's a couple thoughts that the field has established what drives this phenotypic heterogeneity. The first one is environmental influences, just such as diet or caffeine intake. Others are kidney injury, subclinically or clinical. And then what has been studied are to the tubular epithelium, either somatic mutations or germline mutations in modifier genes that somehow interact with the pathways that polycystin 1 and polycystin 2 mediate to then drive cystogenesis one way or another. And on a similar axis, epigenetic modifications that either drive gene expressions or silence gene expression within similar pathways have also thought to drive that phenotypic heterogeneity. But within recent years, what has really gotten more attention is that the changes within the cystic microenvironment might also drive phenotypic heterogeneity. And with that, I mean cells that are within the interstitium. So people have studied myofibroblasts extensively and their role in modifying the disease. And more recently, people have looked at the idea that immune cells and their signaling intrinsically or extrinsically with each other or directly with the epithelium could also drive polycystic kidney disease progression. And the idea that the immune system plays a role in PKD at the time point shouldn't have been that foreign, because what is known that while ADPKD is not generally considered an immune disorder, we do have seen that immune cells accumulate in the interstitium of ADPKD end-stage kidneys, and that has been shown in the 80s already. And then there are cytokines that were found to be enriched in cyst fluid or urine obtained from patients. And using urine models of the disease, multiple people have tried and shown that anti-inflammatory therapies do slow cyst progression. Similarly, kidney injury or kidney stones, which both would provoke the activation of the immune system, have been shown to accelerate cyst growth. And genetic deletion of complement, the key mediator between communication of the adaptive and innate immune system, has also been shown to ameliorate PKD when deleted in a murine model. And These were all kind of observational studies, but the first true functional evidence came within the last 3 years, where groups have deleted M2-like macrophages in PKD and slowed the disease progression in a murine model, highlighting that these cells might be promoting disease. But what really hasn't been studied is the role of the adaptive immune cell, or specifically the T cell. And that is something that I had an interest in, and there was some collaboration within the renal division here, where an investigator, Dr. Nemanoff, has studied T cells specifically in the cancer microenvironment. And so when I was recruited to campus or to Anschutz in 2016, that was the endeavor that I wanted to undertake to understand how T cells might drive cyst progression in PKD. I think all of you can appreciate that T cells themselves are pleiotropic and have a very differing function. Here I'm showing their role in terms of cancer, and cancer chosen just because ADPKD has been described as a neoplasia in disguise, because it is sharing a lot of characteristics with cancer, such as sustained proliferation or resistance to apoptosis, genomic instability, and such. And from the cancer field, we know that there are T cells like CD8 T cells that are truly key in driving tumor suppression, either by being cytotoxic directly, like with granzyme or perforin, or by mediating the phenotype and function of M1-like macrophages. And then on the flip side, there are T cells like the T regulatory T cells, through the cytokines that it secretes, that truly drives M2 differentiation as well as immunosuppression, and as a whole drive then tumor progression. But none of these flavors, or even the idea whether T cells matter at all in PKD, have been looked at the time that I started that endeavor. And so to understand the role of T cells in PKD, we utilized a model that I established as a PhD that mimics PKD, or the human PKD, very precisely. It is a knock-in model that mimics the arginine 3277 cysteine mutation. That mutation is a hypomorphic mutation found in patient, meaning it is found in patients homozygously and only presents with disease if you are having both alleles of that mutation, which is atypical for a dominant disease, as you likely can appreciate. But when we knocked in that mutation into the mouse, and I'm showing here 2 different strains, in the Black 6 as well as the BALB/C strain, what you see at 3 months of age, you start seeing these cystic lesions in the kidney compared to wild type. And you likely also appreciate that there is a strain difference. So in the BALB/C background, PKD progressed much more rapidly than in the Black 6 background. The key reasons why that is to date not known and hasn't been really truly well established or studied. But for no matter what, this mutation does mimic the human disease very precisely, and hence is used for preclinical trials consistently within academia as well as industry. And so to understand T cells, we took the kidneys out of these mice at different time points of disease severity, made single cell suspensions from the kidney, and started analyzing them with flow. So very basic in the beginning and descriptive, just to see what we find in regard to T cells. And the first observation we found is that kidney T cell numbers are increased in our PKD model, and that they rise correlatively with disease. So here I'm showing a flow diagram for CD4 as well as CD8. And you can appreciate that the numbers are much larger in the ADPKD model than the wild type control shown here. And when you quantify that, you see that the more severe the disease gets, or as time progresses, the PKD animals have much larger numbers of both T cell subtypes 4 and 8 compared to the wild type, where with age not much actually changed. And looking at immunofluorescence, what we found is that these T cells stained here for CD3 in green localize very specifically to cystic lesions, as shown here with ECAT here in red. And that is not just a random phenomenon. We looked at non-cystic areas within our model as well as cystic areas, comparing black and gray here. And you see that even at very mild disease at 3 months in this black 6 background, all the T cells already are sitting at the cystic lesion. So there seems to be truly some direct effect in terms of what T cells might be doing at the cystic lesion. We looked at the activation status of these T cells, also using flow cytometry and the markers CD44 and CD69. And we found that CD8 T cells specifically are seemingly more activated than CD4 T cells when comparing wild type and ADPKD kidneys. And that is specifically also pronounced in the more rapidly progressive ADPKD model, which is in the BALB-C strain. So then we wanted to look of why they are more activated and recruited to the cystic epithelium. And for that, we did in situ staining for the key cytokines that are recruiting CD8 T cells, which is CXCL9 and CXCL10. And what you can see here that in green are the CD3 cells and in red is the in situ probe for the RNA of either CXCL10 and CXCL9. And you see that there are plentiful red dots in the interstitium of the cystic cells, but not so much in the wild type scenario or within the non-cystic region. And where CXCL9 and CXCL10 is expressed, you also find the green CD3 T cells. And interestingly, at that point, what we found was that CXCL9 and CXCL10 are not solely just expressed in cells within the interstitium, but also by the cystic epithelium itself. There seems to be some active recruitment from that cystic epithelial cell as well. But that was not the case if the cystic epithelium was non-cystic or non-dilated. So given the data that we had, that CD8 T cells are increased in numbers, that they're more activated in our ADPKD model, and that they're seemingly directly recruited to the cystic lesion, we wanted to go beyond this descriptive data and look whether functionally they play any role. And to do so, what we did is we injected 1-month-old ADPKD mice for 8 weeks long with either IgG or anti-CD8 antibody to deplete the CD8 T cells effectively within that mouse. And the depletion is shown here with flow cytometry, looking at CD4 or CD8s. And in the depleted model right here, you see no CD8 T cells. And when we dissected the mice at the end of that 8-week treatment, we could find that by gross anatomy, as well as by H&E cross-sections, the kidneys of treated animals are grossly larger, as well as have larger or more quantitatively larger cystic lesions compared to their control. And to quantify it more than just this visual aspect, what people in the PKD field do is to take the percent kidney weight over the body weight to understand what the severity of the disease is. And looking at that quantification, you can appreciate that anti-CD8 treated animals had larger percent kidney weight, body weight. We also quantified the cyst size as well as the overall number. And we found that the cyst size in anti-CD8 treated animals was significantly larger, but the number of them did not change, which gave us an indicator that the functional role of CD8 T cells in that ADPKD microenvironment likely is around driving cyst growth rather than initiating the event of a new cyst to grow. So at that point with these seminal studies, we had a couple open questions. So we knew that the kidney or the mouse body responds to having the cystic kidney pathology. We saw increased CD8 T cell number. So far, we haven't looked at or know anything about their function, whether they're good or bad. But what we did know is that the CD8 T cells also increased in number, but they also proposed to halt cyst growth. So the question that comes directly to mind and what we ask ourselves at that point is, why does this continue to grow despite having an immune response? So does the cyst escape this immune response? And that is known from the tumor microenvironment, where T cell exhaustion and suppression through immune checkpoint plays a role. And more recently, also metabolic exploration can drive immune exhaustion. And so we wanted to look at these two aspects in ADPKD. So first we looked at immune checkpoints, and they're the most prominently known from the cancer field, which likely all of you know is PD-1 and PD-L1, where typically it regulates autoimmunity, but the tumor cell hijacks this, expresses PD-L1, and then binds to PD-1 on the T cell and inactivates that T cell, which typically, however, halts tumor progression like the CD8 T cell. And so what we found in our kidneys that PD-1 is highly expressed on CD8 T cells, as shown here, compared to wild types, and quantified as well. And what we also found is that the ligand for it is highly enriched on the epithelium, as shown here, with on the macrophages, as shown here for CD64, as well as on the cystic epithelium, as shown here by gating for EpCAM, and appreciating that this checkpoint is engaged in our mouse model. We also looked at patient samples, and so these are end-stage kidneys from ADPKD patients, 3 different ones, and a normal control. And you see by histochemistry staining that PD-1 is largely enriched. And looking at ADPKD cell lines, we also see that PD-L1 is highly expressed compared to cell lines that are wild type for the PKD1 gene. So typically, as people do in the cancer field, is then block this immune checkpoint and see whether it drives a phenotype. And that's exactly what we did based on our exciting preliminary data, that this checkpoint is so enriched in the disease. But disappointingly, what we found just treating BALP-C PKD1 mice for 8 weeks again with anti-PD-1, the phenotype didn't change at all, as you appreciate here on the histology, the kidney weight, body weight, or the cystic index. In cancer, so a lot of times what people do is to block multiple checkpoints because there is some repetition among them. And that was our next step as well. And so one of the other big checkpoints that are done are CTLA-4, which is also highly activated in the cancer field. And so we kind of blindly went in and did a dual treatment. And what you can see is that, as shown previously, PD-L1 alone or PD-1 alone didn't do anything. CTLA-4 alone didn't do anything either, but treating with the combination of both anti-PD-1 and CTLA-4 decreased the cystic burden, and it decreased also the kidney weight, body weight significantly. So studies ongoing now are in our lab is understanding what causes the upregulation of these checkpoints in the cystic epithelium as well as the macrophages, whether the checkpoints that are engaged are limited to these 2 or multiple others, which there are many, many more, and then translating this to the clinic, because as in the cancer field, checkpoint treatment does have certain kidney toxicity, and so directly translating it to a slowly progressive disease is unfortunately not in the cards to do for ADPKD. And then the second thing that I want to show you some preliminary data on that we looked at to understand whether there is a cyst immune escape is the role of immunometabolism or metabolism shifting the function of immune cells. And what is known is that immunometabolism drives T cell exhaustion. And in this figure, there are key pathways that are doing so. One of them being the tryptophan metabolism and kynurenines that are immunosuppressive. Some of them are lactate, as well as glucose metabolism that is known to drive immunosuppression within the cancer field. We have shown, and many others as well, that ADPKD is characterized by dysregulated metabolism. And here we are comparing the kidneys of 3-month and 6-month-old ADPKD to wild type. And you see multiple pathways being dysregulated in the ADPKD kidneys. But interestingly for us was that tryptophan metabolism, which I just mentioned to be immunosuppressive, also popped up. And so we wanted to look at that a little further. And what we found is that the immunosuppressive metabolites like kynurenine and kynureninic acid are all strongly upregulated in ADPKD compared to wild type, as shown here in this heat map, and indicated with the arrows of all metabolites within the tryptophan metabolism. And then here directly quantified being kynurenine, which increases with disease progression comparing ADPKD mice versus wild type. And so looking at the tryptophan pathway, we wanted to focus on IDO1 as the key enzyme because it is predominantly expressed in the kidney and is the key enzyme that can effectively metabolize tryptamine to kynurenine and kynureninic acid. We found IDO1 to be upregulated in our PKD kidneys versus wild type, as shown here by Western blot. And more importantly, IDO1 is highly expressed, here shown in red, in the cystic lesions, as well as in the cells within the interstitium of PKD animals, but barely expressed within the wild type. So that made us excited to test this enzyme and its therapeutic potential in ADPKD. And what we did is we blocked it with a tryptophan analog, which is a 1-MT, which outcompetes the endogenous tryptophan, so not metabolizing it to kynurenine or kynureninic acid, which are the immunosuppressive metabolites. And what we found here by treating for 4 weeks only, that the cystic phenotype of the ADPKD kidney is drastically decreased, as also shown with kidney weight, body weight, cystic number or cystic index and cystic number shown here. The same finding we also had by genetically deleting IDO1, as shown here, where it is an ADPKD model, and then here a dual genetic model of the ADPKD mutation and IDO1 loss. And most interesting to us, what we were studying is the cystic microenvironment. And upon IDO1 loss genetically or by pharmacologically inhibition, we truly shifted the environment to be in favor of halting cyst growth. With that, I mean, we decreased the number of M2-like macrophages, which have been shown in the literature to drive progression. We also increased the number of CD8 T cells, which we shown previously that is a good thing to have more of them because they halt cyst progression. And we downregulated the checkpoint proteins PD-L1, as shown here on the cystic epithelium, or PD-1 on the CD8 T cells. So now our studies go on in understanding which key cell types overexpress IDO1, whether it is the macrophages, the dendritic cells, or the cystic epithelium, what drives the overexpression, and whether IDO1 maybe even has other functions beyond just being the enzyme that metabolizes tryptophan to the immunosuppressive kynurenines. And then looking into clinical translation, because there are FDA-approved IDO1 inhibitors that have been tested in the cancer world, so they might be of potential within ADPKD. So with that, I hoped I showed you that T cell numbers are increased in PKD and that happens correlatively to disease severity, that CD8 T cells play a role in halting cyst growth, and that the microenvironment truly has the feature of immunosuppression, which might be a new therapeutic avenue, and that the dysregulated metabolism, which is a key feature of the ADPKD kidney, might truly also be a driver to drive that immunosuppression or immune cell function to promote cyst growth. There are a lot of uncharted territories that have to be answered when thinking about the adaptive immune response in ADPKD. And so I'm very excited moving these projects further in the field. And with that, I like to thank everybody in my lab, current people as well as past people. We have a great PKD program here in our division, and I have established multiple collaborators throughout my time here. And then I obviously like to thank the division as well as the University of Colorado Department of Medicine for my startup funding, as well as now I am funded through the Outstanding Early Career Scholar Program. And I have funds from the PKD Foundation and the NIDDK as a Career Development Award. And with that, I can take questions.
Thank you, Dr. Hopp. And unfortunately, we have time for only one question. And I don't see any in the chat, but I do have a quick question. So you mentioned that the mortality is usually due to vascular complications. So my question is, are these vascular complications secondary to what's going on in the kidney, or are they direct effects of the mutations?
Excellent question. And both of it is true. So there are definitely plenty of mouse studies now that show a role for both polycystin 1 and polycystin 2 in smooth muscle function, as well as in the endothelium. However, due to the declining kidney function or kidney anatomy and histopathology that you have with these large cysts, your kidney function declines as well, giving additional cardiovascular stress. And so both of these points are likely true to drive these cardiovascular abnormalities.
Okay, thank you very much for a very interesting presentation. So now we're going to move on to our next speaker. If you don't mind, stop sharing your screen, and Dr. Neff, if you wouldn't mind sharing yours. So thank you so much. And in the meantime, I am going to be introducing our next speaker, who is Dr. Preston Neff, and he's an assistant professor in the Division of Allergy and Clinical Immunology. He obtained his BS in biology from CSU and stayed there for his PhD and postdoc, which he continued at UCSF. He joined Andrews Medical Campus in 2018, and he actually proceeded to study the microbiome and its interactions with the immune system. And actually, our paths crossed when studying how HIV impacts the lung microbiome. And so we did some studies together in that arena. Today, his topic is a slight departure for him, as he will discuss the development of an ELASA, which stands for Enzyme-Linked Adapter Sorbate Assay for detection of SARS-CoV-2. And actually, I had to ask him if this was a new technology. I looked it up in Google and I couldn't find anything about it. So it turns out that it's not a typo. That's exactly what he's going to be talking about. And hopefully we will learn something about this technology. So take it away, Dr. Neff.
Great, thanks, Dr. Flores, for that introduction. And yes, I apologize that this might not be well known of what an ELASA is, but that's what I will be jumping into pretty quickly here, so you will learn a little bit. So as Dr. Flores has mentioned, this is kind of a departure from what I originally was working on. And this is kind of stemming from the pandemic when it all happened. If we think back to April or March, when the pandemic was first happening and we were quarantined and at home, there was a couple of opportunities to present some grants. And one of them was for a novel diagnostic in which you're detecting an antigen. So the actual virus itself, not just a molecular test. And I applied for this and received this funding. That was called RADx. So this is my experiments from all of that and my findings. Let me try to minimize my screen over here. So just a little bit of background. We probably already know most of this, but the COVID-19 pandemic happened. It was started around the end of 2019. And by just when I looked up these latest statistics, there had already been in March 453 million cases worldwide with over 6 million deaths. And one thing that I'm going to be talking a lot about is the testing. Prior to the COVID-19 pandemic, viral testing wasn't that available, or just wasn't something that most people were very interested in. And the only times you'd really do a viral test is if you had the flu and you wanted to see if it was in fact the flu. And there was some lateral flow assays available for that. And obviously that has now changed. We do a lot of viral surveillance with testing, and we do to determine if you're positive or not, so we can kind of prevent the testing. And since then, we've already done over 2 trillion tests worldwide and about 800 million in the US alone. And if you look back throughout the cycle of the pandemic, certain times it goes up and down. And lately, we might have said that the pandemic in the United States is diminishing, but worldwide infections continue. I actually just decided to pull up a slide just to see what the infections rates. This was about a week ago. And you can see worldwide, we seem to have peaked, and then we were going down. I did update this, but I didn't want to change my slide. And this is still continuing to go up. And some of the news this morning says that there might be a new variant. So the pandemic is still occurring, and viral testing, especially worldwide, is still something very interesting. So talking about the available tests that are available is that now we're probably very familiar with the molecular tests, which detect genomic properties using mostly PCR. And then there's also the antigen ones. And this is what I'll be more talking about in developing a more novel-based antigen. And a couple points about where we were at the antigen tests was the first ones that were developed actually used an antibody, and that antibody was originally against the original SARS-CoV-1. And a couple issues with that was that while it detected SARS-CoV-2, it was not specific and led to some issues later on down the road when variants arrived, and then it was not able to detect the variants of SARS-CoV-2. And another thing with this testing that I just want to point out is how much we've spent on this testing. And when we look at molecular, these are a little bit outdated. I think my data was from about January, but as then there had already been 363 million tests in the US and over 500 million outside of molecular specific testing at a value of about $60 in the United States, which averages actually at about 86. That's $31 billion already spent just in the United States. When we compare antigen tests, I actually just saw a brand new one at Walgreens that I didn't have listed here. I have the Illum COVID-19 home test and the Binax Now, which at the time I made these slides about January. I think I should have updated these because there's many more available now, but there is about 460 million tests. They're a little cheaper, but not by much, and they're still pretty expensive. And I know for a fact that I had a slight cold and I wanted to go test myself, and at 30 bucks a piece, it's still pretty cost prohibitive. But so far, we've already spent 13 billion worldwide. Just a couple of advantages and differences between the molecular and the antigen-based tests is it is well known antigen tests are less sensitive to molecular tests, but there are several advantages. I already talked about how cheaper they are. Well, one thing I want to point out is even at $30 a test, that means that people probably don't want to spend that much. But what if they were only in the $1 to $5 range? Then people might test more often. And if they test more often, they can get potentially a better detection because they'll test at the peak of viremia. But generally, they still are cheaper. They're much faster. Some of them can be ran now in under 7 minutes. This also means that they're easier to use, especially because there's less specialized equipment. And some people argue that they actually detect active infection, such as infectivity, and you're being contagious. But you can also bring up some points that you might break up some, picking up some broken up protein that isn't actually contagious. So that's debatable. Now getting into the actual, what is an ELASA? Well, instead of using an antibody, which is the most cost prohibitive part of the ELISA in developing a novel test, I'm going to use an aptamer. And an aptamer is a sequence of nucleic acids, DNA or RNA, that are developed to bind a specific target. And I'll explain how that works in just a second. But because of this, and they're able to bind these targets, they have similar functions to antibodies, but they're much cheaper and much faster to be developed, and they can be developed against non-immunological targets. So something that doesn't induce an immune response, which is really important when we're looking at something like a diagnostic assay, because we might not want to target just what causes an immune response. So some key advantages of aptamers versus antibodies. I already talked about how they're cheaper and quicker, but you can also with aptamers include a negative selection. So for example, in our case, and I'll kind of illustrate this, if we're detecting SARS-CoV-2, we can negatively select against the common cold viruses, which then allows us to select against closely related targets. I already talked about the cost in general of developing, but also the scale-up cost is much more simple. You just manufacture more. They're much smaller than antibodies, which allows for a lot of interesting things. You can have more aptamer per assay, and you can have different types of aptamers in an assay, which give you some unique ways of using them. They're also temperature and pH resistant compared to antibodies, which are sensitive. That allows you to store them at ambient temperatures, which means no refrigeration, and you can use these worldwide where refrigeration isn't as available. There's also no variation between batch to batch. And I say that with a key, it does depend on who produces it, but once you have the sequence, all you need to do is manufacture that sequence right away. You don't have to mass produce them as you do in antibodies. And then lastly, you can chemically modify them really easily, which you can increase affinity and stability and signaling. And I'll even show some examples of this in my talk today, while that's a little bit more difficult with antibodies. So this brings us to the test that we're trying to develop, which is an enzyme-linked aptamer sorbent assay. So it's an ELASA and not an ELISA. But the principles are exactly the same. Start with an immobilization surface. You then add your aptamer instead of your antibody. You then are adding your antigen, but again, it doesn't have to be an antigen because it doesn't have to be immunogenic. So your target protein or target anything. Then you add your secondary aptamer binding a different epitope. You link it to an enzyme, that comes a substrate. And just like an ELISA, you get a gradient of your antigen that turns blue. And one thing that's interesting about using an aptamer on both ends is that instead of with an antibody where you have to generate an antibody in one animal and then a second antibody in another animal, and then you have to compare those over and over again until you get a good pair, with aptamers, you get hundreds right off the base. And that allows you to select the best aptamer pairs for different epitope binding or just for a sandwich pair as I'm describing here. And that you can also use multiple aptamers, I already talked about that, and we can quickly modify them, which is especially important when we're trying to target something like SARS-CoV-2, and we want to look at different variants. So now we want to look at developing an aptamer against SARS-CoV-2, and there's lots of different targets we could do. And if we look at SARS-CoV-2, we can see there's many proteins that comprise the structure of it, including S, which probably many of us are familiar with, that's what most of the antibody treatments target. The N protein, very structural. There's also membrane protein, and then the envelope protein. Well, the S protein, while there's 40 to 70 S protein trimers per virion, the N protein has over 1,000, making it the most abundant protein in SARS-CoV-2, and it has the least variation across viral strains, which makes it the most ideal target for a diagnostic assay. So that being said, we chose to target SARS-CoV-2 N protein. I want to point out that we started with recombinant protein and specifically that they were made in human cells. This is essential because post-modification of the protein can cause our targeting to be different. So if we made them in insects or even in monkeys or something like that, the post-modifications might be specific for the aptamers, and that's not what we want. We want the protein that's going to be produced in a person during an active infection. That also brings up the point of that when you're developing your aptamer, you want to make sure that your buffer is going to be very similar to what we're going to be having our virus in. So we created a buffer matrix that we're going to be selecting our aptamers in that was similar to what we would be using to inactivate the virus. So we have detergents in there, and we also have denaturing reagents such as high salt. That's also very specific for N protein, which can be oligomerized, and we don't want that. We want to break it apart, and at high salt, that will keep them in individuals. So running through the Celix procedure, which is a systematic evolution of ligands by exponential enrichment, how this works You get your target protein or SARS-CoV-2 N protein. We add an aptamer library, and this library can also be very specific, and you can actually develop it. And I'll point out one thing that we made that was unique with our library, but you combine them together. The ones that bind, you keep, you wash everything out, you then elute, collect everything. Then this is where we do our negative selection against our counter targets. And the ones I had available at the time were MERS, and then 3 of the common cold coronaviruses. So we're making it highly specific. Then if we're done, we can collect it, or we can repeat the cycle many, many times. We did 14 rounds. This allowed us to produce many aptamers. I'm only going to show you the top 5 as per affinity. These ones which have the highest affinity in the binding of the SARS-CoV-2 protein, which was a KD of about 10. If you ask me, that is comparable to other aptamers that were developed against SARS-CoV-1, maybe a little bit better. Theirs is only 4, and even to other antibodies that were already being used for SARS-CoV-2 had an affinity of about 12. But when you also look at ours, the delta G is how stable the molecule is, and you want this number to be low. And we had a very low number, as you can see, for all of these, which was even better than some of the previously developed ones against SARS-CoV-1. So great. Now we have these 5 aptamers. What you can see is just when we look at these 5, all of them have different shapes and different sequences. So which one is the best is the next thing. Well, the easiest way to test that is, instead jumping into a sandwich, which we would need 2 aptamers, is just to do a direct ELISA, or ELASA in our case. And where in this case we have our surface, we put our target protein directly to the plate. Then we test all of our aptamers, and we have hundreds, but we actually chose the top 24. As I mentioned, I'm only going to talk about the top 5 or so, just for a point. And then what we also did was we biotinylated these so that we combine them to a strep avid and HRP. Makes it very straightforward to test through them all. And lo and behold, we get results. Most of my results I'm going to be presenting to are just direct reads from the ELISA plate reader, which is optical density. And in this example of this direct ELISA, which is the results from the very first one we did, if we compare the antibody, which was commercially developed and was originally against SARS-CoV-1, compared to all of ours, when we put recombinant protein on our direct ELISA, we get a really nice signal. And in fact, even on our first try, we were already getting some of them that were detecting all the way down to 625 picograms. So I talked about how fast these can go. We received funding in July, and just 3 months later, we already had a successful ELISA. So much faster than most antibodies, which might have still been in production. The next thing we wanted to do, making sure with this direct ELISA format, was making sure that they are indeed specific. So in this case, we put 200 nanograms, a high concentration of all of the proteins we had from all of the different viruses. And this is our original virus that we had made in-house versus 4 different of the common cold coronaviruses. And I want to point out that one of these was not in the selection, and you can see a little bit of signal. I'll talk a little bit more about that. And then MERS. And when we compare, we can see that the monoclonal antibody, highly specific as you would think, are both aptamers. I'm just going to talk about the first 2, very specific to the wild type virus. There is a little bit of signal to NL63, which has the most homology to SARS-CoV-2. But when we go to lower concentrations that are more likely to be seen in an actual test. And I do get this question a lot. How much virus do you expect to see in a patient sample? And I actually don't know, but I did do a lot of literature look on that, and it seems somewhere between 10 and 1,000 picograms. So this is way more than that, than what we would see. So this background signal does disappear after we get below 50 nanograms of protein. And then I wanted to point out our SARS-CoV-2 targets, but we didn't include the OC42. We just didn't have it available when we did the sequencing. So we do see this as a binding product. It also clears up when we go to lower concentrations of protein, but I want to point out that if you don't select against certain ones, you can then get a positive signal. What's also interesting is that some of the aptamers, and unfortunately for both this case, AP1 and APN2 both had some background here, but some of our other ones that we had that we tested through all the way to 24 do not have this binding, which then allows you to potentially use that as it might be binding a different epitope. So, but our real goal is not a direct sandwich. And the reason why is because it's very difficult to put viral protein what that's not recombinant, but as the whole protein on a plate. So what we need to do is develop a full sandwich. And in this case, we have the exact same thing I showed before. We have these strepavidin plates. We got an aptamer. So we're going to biotinylate those just to make this really easy so that our aptamers will bind them, because how else are you going to bind a oligo to a plate and maintain its shape? So we tried biotinylated, making sure this, because we also biotinylated our aptamer to bind to our strepavidin, we wanted to block all the excess biotin. And lo and behold, I will give you the results are that it doesn't work. Unfortunately, when we look at these results, when we look at 200 nanograms of recombinant protein, all of our aptamers didn't work at all. When we use the monoclonal antibody as the capture, it does. And then the detection didn't seem to matter as long as we use the monoclonal antibody as the capture. So unfortunately, it's not as straightforward as just biotinylating an aptamer, putting it on a strepavidin plate, when you also have a biotinylated strepavidin HRP concentration. So we had these inconsistent high background with low detection problems. And as already mentioned, it could be the biotin competition. Maybe it's steric hindrance, where when we're pulling the aptamers and the antigen together, because the aptamers are so small that the protein itself is hitting the plates, that's possible. Or the aptamers, when they're binding the plate, are not keeping their secondary shape. So should we try a different immobilization surface? We also tried that. We tried nitrocellulose paper, we tried glass, we tried all sorts of stuff, and we always had the same issue. So taking a step back, we decided, well, let's still move forward, but this time, let's just keep the antibody on the plate. So it's not a true full ELASA, but it's more of a hybrid ELASA. And when we do this and we make this protein, then the aptamer is our detection, which is binding our enzyme just the same way. Lo and behold, it works beautifully. And this time, instead of just showing you raw ODs, I actually showed you the picture of when we did this. This top one is the monoclonal antibody. And remember that this entire plate as capture is a monoclonal antibody. And we have two different types of pairs because we can use different ones. And this is a monoclonal antibody coming with the kit that we bought for this particular plate. This is a scramble. So one of these aptamers, but we messed up the sequence on purpose and it doesn't work. It looks great. And then you can see we get a nice signal with AP1, 2, 3, 4, and 5. And then we tried a second pair of antibodies, and you actually see our aptamers are working even better. So an aptamer with a monoclonal antibody works really well. We obviously repeated this many, many times and then tried to improve it. And after improving it, we got the detection. And again, I'm just going to show AP1 and AP2, looking at the ODs and the variability of the protein. We can look at that when we look at picograms of protein, we can detect all the way down to under 50 really consistently, and even in some cases lower than that. So really good. We're already getting into that range of detection that we would need for this to be a diagnostic test. So that being said, how does doing recombinant protein look on the detection of inactivated virus? So instead of doing recombinant protein, we put an activated virus in this format. And what we found, very similar results, almost works exactly the same. But the amount of inactivated virus is based off of PCR. We don't get as much of a signal. And I should have put the conversion on here to say, how many picograms did we expect this amount of virus to be? And I believe I didn't have that on here, but it was, the virus is less than a femtomole. So when you do it, it should be detecting somewhere around the 250,000 to 125,000 range. And when you look at AP1 and 2, we can see that they are detecting to that range. Looks great. Obviously, again, we repeat this many times. And that was an N of 1, but this is an N of many. And then we can get the same kind of thing where we're getting a detection all the way as low as around 125,000 to 250,000 copies of SARS coronavirus 2 inactivated virus. Now, how does that compare to the other tests that I mentioned that are also looking at antigen? Well, the ILUM is about 1 million per mil, so we're better than that. And the Binax now is about the exact same that we got to. When we look at viral variants, which is an interesting avenue because, as I mentioned, we can quickly modify our aptamers in our test. And since we have hundreds, some may bind to other viral variants more than the original ones. And so, for example, when we compare APN1, APN2, APN7, or the monoclonal antibody pair that we have, we can look at which ones do they bind well. This SARS-CoV-2 is the one we made in-house, is actually made by Brian Geis, our collaborator. This is the alpha version. Then we tried beta, delta, gamma, and Omicron. And as you can see, all of them bind both the in-house and our alpha version pretty well. Interestingly, was AP7 doesn't like our wild type version as much as it does alpha. So maybe there's something interesting with that. When we look at beta, we actually think that now we're looking back to this and we try to repeat these results, and we think our beta stock wasn't very good. So this might not be true that it wasn't binding beta, because in every case it wasn't binding beta. So we're going to go back and remake it. But then when we start to look at something like delta, APN1 binds it, APN2 does, but APN7 doesn't, and the monoclonal antibody doesn't. Same kind of thing with Omicron, is that you get a differential binding. APN1 will bind Omicron, APN2 doesn't. APN7 really binds Omicron. And if you guys recall, Omicron was one of the ones that was most susceptible to being able to evade on a lot of the antigen-based kits. So if we could, in a way, take AP7 and apply that to some of those antigen kits that are commercially available, we might be able to fix that problem of them not being able to detect this particular variant. So next thing we want to test is moving forward. We got this successful kit. Will it work in different human samples? And this is a big question, because what we wanted to do was to say, instead of doing that invasive naropharyngeal swab that feels like you're scratching your brain, can you do it something as simple as saliva? And as you probably are already looking at this slide, when we do our PBS buffer, we get a very nice read on our APN1, is just my example here. But when we add it into the saliva, we immediately get some kind of inhibition. Interestingly, this is also true for monoclonal antibodies. So it's definitely a saliva kind of competition or something. And I will tell you, we spent a lot of time, I don't have too much time to go through all the data today, on trying to figure this out, whether it was mucins or deactivating enzymes, all sorts of stuff. These are oligonucleotides. They could be sensitive to DNases, but unfortunately, we didn't have much success with this. So I'm just pointing out that we did try, and I spent a lot of time on that. So now going back to our last and kind of more interesting thing, is that we wanted to really truly develop a full sandwich SARS-CoV-2. And we troubleshooted the capture portion of this for a really long time, and using the biotin just didn't work. Well, I was at first really discouraged because somebody else came out and they fixed this problem. And they found an aptamer that works in a sandwich format. And we look at how they did it. They did it slightly different, where they use a thiolated capture aptamer on a malamide plate. And then all of a sudden everything works. And then if you look, they're looking at SARS-CoV-2. Obviously, we're looking at N, so we have some differentiation from there. But otherwise, everything is the same, where they're using an aptamer pair here for the capture and for their detection. So exactly what we wanted to do. So simple enough. What happens if we thiolate our aptamers? And this was done in collaboration with one of our groups that's working with us through the Radix Reach program, which I'll talk about also in just a second. And they ran this, and they just shared this results with me last week. And what looks very exciting is, I will go through these data. This is the amount of protein. This is nanograms, so a very, very large amount. And so we get different concentrations. This is a capture going across on this row. And then this is the detection. Also the same down here is capture and detection. This is one pair of monoclonal antibodies. This is another pair. And then we use that same monoclonal antibody as capture, as I've shown before. And we see that it works pretty well with our aptamer 1. For some reason, it didn't work with aptamer 2, but it works with aptamer 1. Now we're going to get to our interesting data. Using our aptamers as capture. When we use AP1, we have to find a good pair. It doesn't seem to work very well with itself. It doesn't seem to like AP2. You start to see a signal with AP3, and you start to see one with AP4. You get a similar kind of thing with AP2, doesn't like AP1, doesn't like itself, but AP3 and 4. 3 doesn't work at all very well for a capture. 4 seems to work really well, which is really exciting. And although this is an N01, we might have been able to develop a full sandwich pair, which we just ordered all reagents and we'll be following up with. So, and that just says that we're down to already 50 nanograms, but do need some approvals. This brings me to my conclusions on this talk. We have selected novel aptamers with high stability and affinity. They're very selective, and our signal is 25 times higher than background. We developed a hybrid ELISA. That's where we had most of our success. And we got it to be very sensitive at less than 50 picograms of recombinant protein and under 250,000 copies of viral protein, or a virus, I mean. And then most recently, we developed a full sandwich that can detect recombinant protein. This was done also through external evaluations, which is very interesting. All of these reagents have been sent to an external lab and they reproduced our results. Never in my life where I do something and you send it off and another lab reproduces it is a wonderful feeling. It was really great to have that happen. So in our total conclusion, we've developed a standalone ELASA. We're hoping to make that into a full sandwich, which could be its own kit. And then mostly what we've done is developed aptamers that are reagents that are replaced to capture and detection antibodies and other standard antigen-based tests. With that, I'd like to make my acknowledgements. I mentioned already that this was funded through the RADx Tech program, which was under the guidance of Spark Reach, and they provided a supervisory team, which was Richard Duke, Floyd Taub, and Stephen Wolp. I specifically, in my lab, want to thank Richard. When he first joined, I made him spit in a tube for about 3 weeks straight, just to try to solve that saliva problem, which we never did. My other team members, Mila Sakara, who's helping me on the business side, and Dr. Thomas Campbell, who's our physician advisor. Brian gave us all of the stocks, and Chuck Henry is our technical advisor. And I also have to give a shout out to Brent, who's always guiding me as my KO1 mentor. And with that, I'll take any questions.
Thank you, Dr. Neff. We have time for one question, and I don't see any in the chat, but I do have a question actually, and you kind of answered it towards the end. I was wondering about the stability of these aptamers in a biological fluid, right? Especially saliva, because saliva is loaded with nucleases, for example. Right. And so you showed something, obviously, that saliva, when you did the first iteration, it just didn't work until you thiolated the aptamer. Have you done the new iteration with the thiolation with saliva as well?
We haven't tried the thiolated, but we did try other protective mechanisms. The I can't think of the name. The phosphodiol backbone. Yeah, we tried all of those. You know, what we really did, to answer your question more specifically, was is it that the breakdown of our aptamers in saliva? So the easy test was we put our aptamers in saliva and then we checked 10 minutes later. 10 minutes later, the majority of our aptamers were still together. They weren't degrading. If you left it for longer than an hour, they do start to degrade. But our saliva tests are really short. We're taking in saliva, we're putting them in the aptamer. They're only in the aptamer for about 30 minutes at tops. And we couldn't get to work. And what was interesting was the antibodies, which obviously don't degrade as much in saliva, just don't work as well. And it just turns out that the saliva is a really complex matrix. We looked at lots of different groups and trying to figure out why. And they say it's the mucin. Some say, you know, the bigger problem that, and I just talked to a group last week about this, is you get a saliva from one person who hasn't had any coffee or any beverages in the morning. You get saliva from somebody who has coffee. It's different than somebody who had breakfast. And all three of those are all different, and you need a test that can really do it. And that's why partly the nasopharyngeal swab, even though you would think that there's an equal amount of virus between the two, has been a little bit more reliable for the diagnostics.
Okay, well, thank you. Even though there's a lot of mucin present there too, so.
Yeah, right. Yeah, I think it's just more consistency is what I really get.
Well, so thank you, Dr. Neff, for, again, a very instructional presentation. I learned a lot. So I'm sure a lot of people in the audience also learned a lot. So thank you very much. And thank you, Dr. Hopp, as well, if you're still on. And so thank you for all of the participants attending here. So thank you very much and have a great rest of the day. Bye.
Great.
Thank you. Bye.